Hey everybody, and welcome to chapter number 14. We're talking about smell today, and we are entering into our final stretch of the book and of our class, because chapter 14 and chapter 15 are about our chemical senses. They are about smell and taste, and as the name suggests, those are based on chemical properties that we are able to detect in our environment around us. That is very different than the unit that we just kind of got out of, where we were talking about hearing, we're talking about vestibulation, we're talking about touch, and all of those operate based on mechanoreceptors, right? So we're talking about these cells, these neurons, that if you bend them, if you distort them, if you push them, if you vibrate them, they send out action potentials as a response. These two chapters, we're talking about neurons that, when they detect a specific kind of chemical signature, send that over to the brain. But I don't want to tease you too much about it. Let's just go ahead and get right into it. So we're going to be talking today about what smell is. I'm going to run you through the anatomy of the nose and of the olfactory epithelium. We'll talk a little bit about how that stuff gets from here, from your nose, over to your brain. It's not that long a trip, as you can probably see. We'll talk a little bit about how humans are able to smell, some theories about smell perception, and how we can communicate through smell, or if we can. Okay, so we got a lot to talk about. Let's get into it. The videos that I have for you here, I don't think that any of them are particularly crucial to understanding the course material, but I think like these first two are kind of fun. They're kind of fun videos. The human ability to smell is kind of a fun thing where they kind of like compare it against other species. COVID and smell. I included these two pieces down here because some of you may know, and we'll talk about this a little bit as we go on, that COVID, if you've ever had COVID before, one of the like kind of defining symptoms of it is that you lose your sense of smell. And that changes the way you taste things. And if that is you or anybody that you know, these videos kind of explain why that happens. This one down here, this New York Times piece, they have a great podcast episode that kind of unpacks that and kind of talks about it, the importance of smell, and how a lot of people didn't realize how much they love smelling things until they lose the ability to do it. Okay, so smell, like I mentioned, is different than all the other senses we've covered so far this semester. If you think about vision, what is vision? Vision was that we have these photoreceptors in the back of our eye that are sensitive to the electromagnetic spectrum. And that if we come across light that is between 400 nanometers and 700 nanometers, that we can actually detect that with our photoreceptors. That's how vision works. Hearing, that's mechanoreceptor stuff, right? That is that we are detecting some vibration or some displacement in the medium around us. Orientation, that's detecting head movement. So if you move your head, we have receptors in our inner ear that are responding to that. Those mechanoreceptors are bending and swaying with that fluid. Then we got touch, which is, again, mechanoreceptors, where we are detecting deformations in our skin, deformations in our flesh. So if you poke, if you prod, if you bend or rub or scratch, that we detect that because the cells in our epidermis are being activated. They are creating action potentials because the structure of the cells are being smushed. And this brings us to smell. Smell and taste are very similar. Smell is the detection of chemicals in the air. And that's all that it is. It's just that we are detecting chemicals in the air around us. We take a deep breath in, and then we detect if that air that we breathed in had any kind of chemicals that we might recognize or that might be important to us. Let that sink in for a second. That whenever you're smelling something, so think about something that you smelled that was gross recently. What your body is doing. And I apologize, this is gross, but this is your body. You've been doing it for 20-something years now. Whenever you breathe in something, let's say it's something stinky like a rotten eggs, let's say. I know you can think of something worse, but let's say it's rotten eggs. You hid something. You hid some eggs on Easter months ago, and now you're just now finding it in your yard, and you smell it, and it's disgusting. And the reason why you're able to smell it is because little particles of those rotten eggs are floating about in the air around you. They are floating about in space, in the atmosphere. And whenever you smell them, whenever you smell that rotten egg, you're taking that breath in, and those little particles are they're riding that oxygen. They're going to go up into your nose, and they're going to cling to a mucous membrane that is up in the top of your sinus cavities. And that is how you were able to detect it. So whenever you smell something gross, it is because that gross thing has actually, a little piece of it has entered your body. Yeah, it's disgusting. Okay, so let's talk about how this disgusting sense works. When we talk about odors are just any kind of chemical stimulus that we have turned into a sensation. In other words, it is a stimulus that we have translated into a signal. So that is odor. So odor is that translation. And odorant is going to be the molecule that is translated. So an odorant is going to be the molecule that has a very specific kind of physiochemical property that we are going to recognize, or that we are going to translate into an odor. This gives you an idea here, for example, of how I'm trying to remember exactly what the textbook is trying to point out when it brings this stuff up. That when we're talking about specific kinds of smells, that what we're detecting is the structure of that chemical. And so the difference between menthol and a green pepper are going to be relying not just on like, well, what kind of chemical signatures is it? Is it hydrogen? Is it oxygen? Is it something else? Really what we're responding to is the structure of it and how those things are put together and how those things combine into their own unique kind of signature. So in order for us to be able to smell something, because not everything is smellable, right? Like carbon monoxide is not easily smellable. In order for us to be able to smell it, it needs to be volatile. So it needs to be able to float through the air. It needs to be small. And it needs to be hydrophobic, which means that it is repellent to water. Carbon monoxide is volatile. It is small, but it is not hydrophobic, which is part of the reason why it is difficult to perceive. So let's talk about the anatomy then. So you know that it's in your nose, but what you may not know is that the nose is, what you may not knows, what you may not knows is that the primary function of the nose is not actually to smell things. Really, your nose is like a really good humidifier and you know this because you're New Englanders, right? Like, you know what it's like to go out, let's say in November, December, January, February, March, and a good part of April. You go outside and you take a deep breath in with your nose. That can hurt, right? Because that really crisp, cold air, your nose is meant to kind of help stop that from happening, that cold shock. Because it'd be harmful for your lungs to take in extremely dry, cold air. That can actually hurt your lungs. And again, you know this because you've taken a deep breath in on a nice, chill winter morning. So your nose is actually meant to draw air in, but humidify things while it does that, to keep it warmer before it makes contact with your lungs, and to also to filter things out. Now, your nose has lots of tiny hairs in it. You're sensitive about it. You don't want anybody else to know it. That's why you don't look up like that, because you're worried that people are going to see all the gnarly, disgusting hairs that are in there. But those hairs are a great filter. They stop a lot of particles from making their way into your lungs. They stop all that dust, all of that dirt. So it's good that you have those nose hairs in there. So that's what our nose is for. Now, residing up above that, we have an olfactory cleft. And in this olfactory cleft, it's like this hollow space that's up there. That's where we're going to have our primary sensory organ. Our primary sensory organ is the olfactory epithelium. And so you can think of it very much like the retina of your nose. You know, I love analogies like that, right? Whenever I was talking about hearing, for example, I made a point that the cochlea is kind of like the retina of hearing. It is the primary sensory organ. It is really what changes that physical stimulus that is an objective, it has objective properties, empirical properties that you and your friend and your mom can measure and agree with. The olfactory epithelium is going to take that physical stimulus and turn it into a subjective stimulus. It's going to turn it into something that your brain can understand. And so even if you, your friend, and your mom are measuring these odorants, and you can all agree that they are all odorants and that they are all a specific kind of smell, the three of you may disagree on if that smell is pleasant or not, right? Because that is subjective. That is something that we can't really directly measure. That is something that is personal to you as part of what we call the qualia. And I don't know if I've actually even used that word all semester. I talk about it a little bit in cognitive psychology, but qualia is like your subjective experience of that stimulus. So we're talking about psychophysics. To go back from chapter 1, we're talking about psychophysics, which is the relationship between the objective world and the subjective world. The olfactory epithelium is what's going to help you convert that into something that your brain can understand. So here is the olfactory cleft that you see up here. And so most air that you bring in is just going to go straight, through your nose like that. But we got a lot of space up here, a lot of boogers, a lot of hairs. We don't like to think about it. We don't want to know exactly what's up there because we think it's gross. But that is where your olfactory epithelium is. And this is actually right below where your eyes would be. And so let me just draw the eye right there. And so you can think about how high up that is, right? Like that's pretty high up in here. And it's mostly porous and kind of empty, which means that it's very vulnerable, which we're going to talk about in just a moment. Because sometimes when people get hit in the face, it can actually damage your ability to smell. So we got the olfactory epithelium. It's right up here. We got the olfactory bulb. That's part of, I think it's safe to say you can call that part of your brain. Yeah. So you can see exactly what I mean, where it's like right next to his eyes. All right, so the olfactory epithelium, you can think of it as like the cochlea of smell. There are 3 types of cells here. And if you watch that COVID video that I mentioned in the very beginning of the chapter, they talk about the supporting cells a pretty good bit. Do you want to guess what the supporting cells do in your olfactory epithelium? If your guess was that the supporting cells help support other cells, then congratulations, I will give you a gold star because you're absolutely right. That supporting cells really are going to support the olfactory sensory neurons. The olfactory sensory neurons are really kind of like the rods and cones of smelling, if you want to think about it that way. They are going to be what really transduce they are going to be what detect, what kind of grab a hold of the odorants in the air and recognize it, and then based on the pattern of that odorant, turn it into an electrical signal that we call an action potential, and then communicate that up to the brain. The basal cells, don't worry too much about the basal cells. They're just kind of like precursor cells that kind of help support the olfactory sensory neurons as well. The most important ones here are going to be the OSNs, the olfactory sensory neurons, but it is also important to say that if you have COVID, for example, and COVID can target the supporting cells here, and the supporting cells really kind of help keep the olfactory sensory neurons alive, keep them healthy, keep them active, keep them clean. If you lose those, then you can also lose your ability to smell, which is what we see with COVID and things like that. Those olfactory sensory neurons are going to be in this mucous membrane that is. Sorry, I forgot to put a timer on to know exactly how long I've been yelling at you. It's going to be in this mucus, and that mucus is important because when we take our deep breath and those odorants come in, they're going to get stuck in that mucus, and that helps us kind of helps the OSNs quote unquote grab a hold of those cells. So here we have hair cells, and these hair cells are different than the other hair cells we talked about. We talked about hair cells for hearing. We talked about hair cells for vestibulation. They are very different than that. Those hair cells were mechanoreceptors, which were to say that they basically were dendrites that if you push them in one direction or the other would cause the cell to fire an action potential. That is not how these hairs work. The cilium, and the plural of that is the cilia, which you can see right here. These are dendrites that are hanging off of the olfactory sensory neurons. And those dendrites are essentially what lock on to these odorants that come in. So they're kind of the first step in the road to transduction. That whenever we take a deep breath in, that these cilia, these dendrites, are going to be latching on to incoming particles. And then from there, as we're going to talk about when we talk about shape pattern theory, depending on the shape and the structure of that odorant, that's going to affect how these olfactory sensory neurons respond. How are they going to fire faster? Are they going to fire slower? Are they going to fire the same? A quick note about up here, you're going to see that inside the olfactory bulb that we have lots of different kinds of cells, like the granule cells, the mitral cells. You do not need to worry about that for this class. It's not going to be on the exam, and good chance you'll never, ever, 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 ever need to know about those things, because I've been teaching this class and I've been doing research in the field of taste and to a lesser extent smell, and I've never never thought about these cells, so I don't think that you'll have to either, unless you get a PhD specifically in this stuff like I did, and even then you still may not need to know about it. Okay, let's talk about the roadmap to the brain. Let's talk about what happens now that we have these cells transducing these chemicals. Now that we have these cells that are firing and communicating their signals, what they have translated those odorants to be, let's see what happens on the way to the brain. So from there, we're going to go to the olfactory nerve, which is what you're seeing right here. So we got the OSNs, we have these olfactory sensory neurons. They are going to communicate their findings up through this cribriform plate. So it's going to go through this very porous kind of cartilage that is very easy to break. When it moves up there, it's going to go into our and I should also point out, these right here, this is the olfactory nerves. So the olfactory nerve is very similar to the auditory nerve, very similar to the optic nerve, in the sense that they go from our primary sensory organ straight to the brain. That's not a very long trip, as you can see here. It goes basically straight through this cribriform plate, and that's pretty much it. From there, the olfactory bulb is going to further kind of interpret these signals and then communicate that to the brain. Okay, so the olfactory nerves, those are going to be those bundles of axons that you just saw that go through the cribriform plate. From there, they go to the olfactory bulb. The olfactory bulb is part of the brain. It doesn't get a lot of love though, because it looks kind of weird. It actually looks kind of like this tubular thing, and I actually have found that whenever I see people who try to like model the brain, like as you can see here, this brain does not have olfactory bulbs on it, which is disrespectful to the sense of smell. But the olfactory bulb is these kind of like thin tendrils that are blueberry size, as the book refers to it, just above where the nose is, and that is going to process some of these signals and then escort them later to the brain. We do have two olfactory bulbs, both on the left and right hemisphere. If you plug up your one nose right here, you take a deep breath in, that is going to be ipsilateral. In other words, it is not going to go to the other side of the brain like touch does, and like hearing does, and like vision does. That if you plug your nose and you smell something, that is going to stay in that side of your brain. From there, we're going to go through, I mentioned through the glomeruli, but that's part of the olfactory bulb. But from there, it's going to make its way over to the primary olfactory cortex, which is going to be where a lot of that is going to be further processed so that we can understand what it is that we're smelling. Here's the interesting thing about the primary olfactory cortex. This area is not, it's actually a collection of areas. It's like a chain of areas. It's not like vision, where with vision we had the V1, and it's not like hearing, where we had the olfactory cortex, the A1. It's not like touch, where we had the somatosensory cortex. The primary olfactory cortex is it's a collection of other pieces of anatomy, and those are the amygdala, the parahippocampal gyrus, and the entorhinal cortex. Now, if you've taken cognitive psychology, two of these should jump out at you. Two of these should be like, hey, those sound familiar. And that's the amygdala and the parahippocampal gyrus. The amygdala is what a lot of people call the seat of emotion. And I don't know why the word seat is so commonly used here. It's kind of like when people talk about the mitochondria and they say, oh, that's the powerhouse of the cell. And it makes me think, like, why does everyone use the word powerhouse? Like, you never hear the word powerhouse unless someone's talking about the mitochondria. But when people talk about the amygdala, they say it's the seat of emotion. So we got some emotion going on here in our primary olfactory cortex. The parahippocampal gyrus, that's going to be used in memory. It's related to the hippocampus. And then we got the entorhinal cortex. And so you may be thinking, like, okay, well, I've heard that smell is highly, I don't know, maybe I'm projecting onto you, but whenever I hear people talk about smell, my ears perk up, which is weird. You would think that my nose would perk up. When I hear people talk about smell, usually, like, a lot of times people will be wrong about that stuff, and so that's kind of why I'm always like, okay, what are they about to say about smell? And one of the things I almost always hear is that smell, something about memory, right? Like, we know that smell and memory seem to they work well together. Well, this is part of the reason why, is because one of the areas responsible for interpreting those smell signals is directly related to memory. Another reason why we can have really strong emotional connections to smell, specifically disgust, like whenever you smell something and it makes you want to throw up, that's because the amygdala is part of that seat of emotion that is going to register whenever you think something is gross or disgusting. And then we have the entorhinal cortex, and the entorhinal cortex I feel like is not one that I was as familiar with. It doesn't come up in other areas of psychology the same way that the amygdala and the hippocampus do, or the parahippocampus. I should be specific about that. The entorhinal cortex is a kind of evolutionarily old, ancient area of our brain. It's deeper than some of these other areas, and it's going to be used for helping kind of maintain sensory associations. And so what do I mean by that? Like, if you think about like classical conditioning, where you're kind of creating associations between, two things, the entorhinal cortex is going to be used heavily there, where we're going to be using that to help kind of make these associations. And so, hopefully that gives you something to think about. That when we talk about our sense of smell, maybe the reason why it feels a little bit different than some of the others is because, on one hand, it could be more emotional because the primary perceptual region of the brain that's responsible for processing the stuff is the amygdala. And it could be tied to memory because the parahippocampal gyrus. And it could be built around associations we formed because of the entorhinal cortex. You can see those areas here. So we got the entorhinal cortex down here. We got the amygdala here. That amygdala actually looks pretty big it's actually a little bit smaller than that. It's actually about the size of an almond. And then we got the parahippocampus and the hippocampus, which are parts right there in your medial temporal lobe. Let's talk about the human ability to smell. Let me take a quick sip of coffee after I smell it. Okay, so every chapter, I always mention to you something about how, like, oh, it's too bad we're doing this online right now, because if it was in person, I'd have this really cool interactive thing for you. This is where you are missing out, because when I'm testing this in the classroom, what I do is I bring a dog to class. I bring one of my dogs. I set out my dogs here because none of them wanted to be here while I talked about smell for some reason. But I'll usually bring one of my dogs to the class, and we do a competition. We see who can smell better, humans or dogs. And you already know the answer, right? You already know that it's dogs. And so we put that up to the test because that is a testable question. And sorry, I feel like I keep my nose is really itchy, and I don't know why, and I feel very self-conscious because, like, I'm like rubbing my nose like I just snorted some cocaine or something like that. Which just to be clear, I have not today. And also to be clear, I have not this week. And to be clear, I have never done that in my life, actually. I just thought it'd be funny to specify that. But kind of interesting. If you do a lot of cocaine, it can actually ruin what's called, it's not called the fulcrum, but what is it called? This cartilage that separates your nostrils. If you do too much cocaine, you can actually erode that, which is really scary, and create essentially one big nostril. Don't do that. And in that, I've always wondered, well, in that case, what happens to the smell if you have one nostril? Is it the case that, like, if you plug up this side and take a breath in, that only that goes to the left hemisphere, or can some of that go over to the right hemisphere because now you no longer have that division? I don't know. All right. Anyways, let's talk about. I'm talking about dogs. So we do a competition, and usually what I will do is I will heat up some meat, usually chicken, and I'll put it in a Ziploc bag so that it gets really, really stinky, and I'll microwave it so that it gets even stinkier, and then I'll take it out in the classroom and I'll hide it somewhere, and I'll have one, volunteer hang out outside the classroom while another student hides this really stinky chicken, and then we'll let them in, and then we'll see where they go. Can they follow their nose to find out where that stinky chicken is hiding? Honestly, humans are better at this than you might think. They're actually pretty good at that. It's just that dogs are even better at doing that. So why is that? It's partly because we have a lot of olfactory sensory neurons, and those olfactory sensory neurons help us kind of discriminate smells, but dogs have over 100 times more than we do. And they have more of their brain dedicated just to smell than we do. I don't have a dissected rat brain to show you, but if you look at a rat's brain, I want to say it's almost half of its brain is dedicated to processing olfaction and smells. And that's very different than humans. As you saw, we just are talking about this little blueberry piece up here, the olfactory bulb, and then we're talking about some of these smaller areas down here. The equivalent of that for a rat would be like this whole back section is just dedicated to smells, which is why rats are really bad at seeing, but also really good at smelling things. This figure right here is a really, really fun experiment where in this in figure A, what you're looking at is that they took this dead bird and they dragged it across a field, and they let this dog loose to see, all right, can this dog follow the scent of this dead animal? And you can see that it can, but not only does it follow it, but it can also kind of weave back and forth. You see its trail is making, it's going all over the place. But that's okay, because this dog can pick up really, 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 really low concentrations of that smell. We humans can smell as long as it's in a high concentration. And so what you see in this figure is a participant who, they have on headphones, they actually have on goggles so they can't see. They are just crawling around on all fours. I guess in this guy's wearing khakis. Rip, right? Good luck getting those grass stains out. But they took some chocolate and they drizzled it through this field to see if this human could follow the scent of chocolate. And you can see they can. The humans can do this, but they have to stay very close on top of that smell or else they'll lose it. Whereas the dog doesn't need to. It can pick up these low concentrations, whereas the human can't really. Here is a really fun look at the different kinds of species and how well they do on how big the olfactory bulb is, or how many neurons they have in their olfactory bulb. And so the higher, generally speaking, the better, and the lower, the worse. And so you can see that male and female humans are pretty low. Women, generally speaking, have a better sense of smell than men. We'll talk about that a little bit in just a moment, why that could be. But I thought this was fun, right? That, for example, that we got rats, and I'm just talking about how good rats are. Well, this is this compared to this. You can see that there's some other species that are even better. I'm trying to remember here that Dasyprocta. What the hell is Dasyprocta? I'm just going to look this up real quick and then show you an example of it. Dasyprocta. Oh yeah, it's called the agouti. I don't know if you've ever seen the agouti. It's from South America. It looks a lot like a, if you can see that. What am I doing? I got a computer right here. Hold on one second. Let me just show it to you. Okay, all right. So this is what I'm talking about. Cute little guys. They look very similar to chinchillas, kind of, but I always felt like they almost look like capybaras, if you know capybaras. And you can see how big they get. Pretty big fella. Look at that. How cute is that? You should check them out on YouTube. They are extremely good at smelling things. I don't know why we don't have, like, I don't know, like bomb sniffing agoutis, you know? All right. Anyways, fun graph to show, you know, the difference between genes that are responsible for smelling across different species. Fun. You can see that humans relatively low compared to non-humans. Look at these dogs. Like 811 genes specifically for olfactory reception compared to humans. And hey, elephants are killing it. Okay, so let's talk about anosmia. So anosmia is the inability to smell. And this can happen for a couple of reasons. It can happen because of some kind of illness, which is what we see with COVID, or it can be from head trauma. And so if you have if you get hit in the face, somebody punches you in the nose, or if you get a baseball that hits you right in the face, that can actually slice through. It ruptures and splits that cribriform plate, and in so doing, it slices right through those neurons, those olfactory nerves, slices right through them, so that the olfactory sensory neurons, they're working just fine, but they can't communicate their findings now over to the olfactory bulb. And I should say specifically, this is for anosmia that's caused by head trauma. The kind of anosmia that's caused by illnesses are generally going to be issues with either the mucous membrane of the olfactory epithelium, or, like I said, the supporting cells of the olfactory epithelium. So, scary stuff. But about 1 in 20 people have it. So there's a good chance that at least one person in our class has some form of anosmia. And in fact, usually whenever I talk about this in the classroom, there's a couple of people who usually will say maybe they got COVID and they just haven't regained their sense of smell yet. So it's not an uncommon thing. But to the point that I made at the very beginning of the semester, you know, like, why is that vision is taught so much? Why is the book so heavily vision focused? Is because if you lose your sense of smell, you can kind of go through life without a lot of help, right? Like if you're non-sighted, if you're blind, you got to have some device to help you get around. Maybe that's the seeing stick, maybe it's a seeing eye dog, maybe it's Braille. For hearing, we need either hearing aids or a cochlear implant. We need to learn sign language or whatever. For smell, if you can't smell, I think society is just like, okay, well, who cares? Yeah, and they don't take it all that seriously, unfortunately. So anosmia is for all smells that you're unable to perceive it. You're unable to detect these incoming signals. But there can be some cases where you can smell some things and not others, and this is called specific anosmia. Not a very imaginative title, I know. How do you like my Westfield mug with Nestor the owl on the front? So specific anosmia is whenever there's one kind of chemical compound that you seem to otherwise not be able to smell. You can smell other things, but not this one specific thing. And probably the most common version of this is a specific anosmia to androstenone. And androstenone is something that people produce in their armpit sweat, and it is also found in pork. And so people who smell this, some people have very strong reactions to it, some people don't really so much. And so most perceive it as smells kind of like piss, smells like BO, like body odor. And some other people say that it smells kind of like a, I don't know, like a sweet fragrance, like a really earthy kind of fragrance. Hard to explain, really. But that is not exactly unpleasant. And interestingly, there are actually sex differences with this. And I have to be careful here because it's kind of hard to talk about because this isn't true for everybody. But generally speaking, if you take a cis, straight, hetero dude and you have him smell another guy's sweat, they will say it's unpleasant. If you have a straight, cis female smelling the exact same sweat, they will rate it as far less unpleasant than men do. Are you with me so far? And you can tell me you disagree, that's fine. Research says otherwise. It's hard to believe, I know. For some of you, like, what do you mean sweat smells good? But this is where it gets really fascinating. If you take that same sweat that straight men do not like, and you present it to a gay man, the gay man will rate that much higher than the straight man does. But you can also see the same thing going on for women who are gay. So if you take the body odor of a woman and you present that to a straight identifying woman, they will say that it smells bad. If you present that to a gay woman, they will say it is not as bad. And some will even say that it's preferable, that they actually kind of like the smell. But just a really interesting, fascinating kind of way that our brain interprets these signals. And so whenever you hear people kind of talk about how like there are biological and physiological differences between sexuality and stuff like that, that's one of the really kind of fascinating examples of this, where it seems to be like biologically that some people just do not like, that smells bad. But for others, it doesn't, and that there might be some interesting kind of hormonal things at play here, creating these differences. All right, so let's. We already talked about this stuff. Yeah, and this is a cribriform plate, where how if you were to get hit in the face really hard, it can fracture this and slice through. these olfactory nerves, so that your olfactory epithelium is working a-okay, perfectly fine, but it can't communicate its findings to the olfactory bulb. All right, let's. I feel like I'm moving fast, but I think it's just because, you know, this one sense we're kind of touching on the greatest hits and talking about kind of the big ideas for all of these things instead of unpacking them like we did for vision and for hearing. And also, I love this stuff, and I already told you, smell and taste are my favorite chapters, and so I feel like I want to talk more and more and more about this, but I know I shouldn't for the sake of time. All right, so there are two big competing theories for how we are able to smell things and perceive them, and I'll just be honest with you. I think pretty much by now most scientists agree that it's shape pattern theory. So I'm going to tell you a little bit about vibration theory also, but really I think that shape pattern theory is the one that is worth spending more time on because it's the more kind of preferred perspective on how smell works. So the theory behind shape pattern theory, or the mechanism behind it, is that in order for you to perceive different scents, what's happening is that those odorants, they have a very particular kind of structure. So let's say that I have a smell for a brownie and it's shaped like that. You know, something really, really boring like that. And so whenever this particular molecule fits into our dendrites, those olfactory sensory neurons, we have recognized, oh yeah, whenever we detect this, it's because there's brownies nearby. Remember what we talked about with the entorhinal cortex and how it helps pair associations? So we recognize that as brownies. But if you come across another chocolatey kind of smell, it's going to have a lot of the same kind of chemical signatures. So it's going to have a very similar kind of structure as well. And it may look something more like this. And I'm making these up, in case that wasn't clear. And what you can see between these two is that they do share some structure. They do share certain kinds of like we have like this long stem, and then we have these two kind of circular orbs, where here we have the stems and we got the orbs, but there's maybe more of them here. And so as the reasoning goes, that if you were to smell chocolate, you might mistake it for brownies because it shares a lot of the same kind of physical signatures that brownie odorants do. And so because of that overlap, we sometimes mistake those scents. So those different scents basically are going to work like a lock and key mechanism, and that is a phrase I feel like I always see people talk about whenever they talk about shape pattern theory, that it's a lock and key mechanism, that we might have a dendrite, and that dendrite has this piece that's shaped perfectly like that. What do you know? Perfectly for part of a chocolate or brownie molecule to slide right into and fit really neatly. And so based on how active, and also let me back up 1 second. Maybe there's a neighboring neuron that's a little bit different, right? So this one, the kind of receptacle is not going to really accommodate these molecules, right? Because this one's looking for something with a more jagged edge, with a more pointy edge. And so for that reason, we have all these different olfactory sensory neurons. They all have their own kind of like receptor shape. And depending on that receptor shape, and depending on the different kind of structures that we are inhaling, these odorants, some of these neurons are going to be more active than others, and some of them are not going to be very active at all. And what we are perceiving, what our brain is understanding, is which of these neurons are active and which ones aren't. Which, if you think about it, is kind of how, to a degree, kind of how vision works. When we talk about the cones, and specifically I'm talking about for color, that some of those cones are going to be active because the light is a specific kind of wavelength, and so on and so forth. All right. Vibration theory is a little bit different. Vibration theory is that we have these molecules. These molecules have a very specific kind of vibrational frequency whenever they latch onto these OSNs. And so for that way, it works a little bit more like hearing, where you have these vibrations that are created from latching onto these cells. And this might also explain why we might have some kind of specific anosmias, because maybe we're experiencing kind of conflated vibrational frequencies, and so we can't tell the difference between these two. I don't know. Like I said, I don't want to go into too much detail here because it gets a little bit more complicated, but it's not really worth going into because scientists these days are not as crazy about vibration theory. So instead, I'm going to be spending a little bit more time talking about shape pattern theory here. So what you're looking at right here, we got odorants. We had the odorant receptors. These are extremely simplified cases, but you can see how you can mix and match these, and that some of these are going to fit more easily into some of these than others. And based on based on which of these cells are active, we are going to perceive it as chocolate or a brownie, or as body odor, or as a flower, or as beef, or whatever. So this is also why we seem to be able to detect so many different kinds of scents. So we only have so many different kinds of olfactory receptor types. We have about my understanding is that we have about 1,000 of them, which means that we have about 1,000 different kinds of receptors here. But why is it the case then that if we have 1,000 receptors, why can we smell more than 1,000 different scents? The reason is because we have mixtures. If you think about this cell right here, or sorry, this odorant right here, it can fit into this, right? But only a piece of it. It can fit into this one back here very nicely. It could even fit into this one back here and probably not be an ideal fit, but can fit too. And so this is how we have these different combinations of receptors firing. And it's that combination that really tells us what it is that we are smelling. So this can also change based on the intensity here, how, what is the concentration of the smell, and how, if you only have a weak concentration, that's why sometimes we mistake things where we smell something like, oh, that smells like beef. And you take another deep breath in and you catch a higher concentration and you're like, oh no, actually that smells like fish. All right, let me take a quick sip of coffee. Actually, I'm gonna take a break, 1 second. All right, so let's talk about differences in olfaction. We already talked a little bit about this, right? Because we were talking about how specific anosmia means that some people smell some odorants one way versus another. And, now that I think about it, this is actually kind of profound, right? Like if you think about vision, we don't have that with vision. We don't have where like, oh yeah, 20% of the people, they see blue as red, like let's set aside cases of color blindness where someone doesn't have a specific kind of cone. As I mentioned earlier when we were talking about sensitivity ratings, that women are more sensitive than men when it comes for olfaction, just in general. And this is especially true during specific phases of the menstrual cycle. Sensitivity isn't actually heightened during pregnancy, even though ask your mama, she'll probably tell you that whenever she was pregnant, she had very specific kind of cravings or certain kind of foods she was not into, right? And so we know that there's something about hormonal cycles that changes the way that our body interprets these chemical signatures. And then this can change with time, not just in terms of, whether, what time, or I'm trying to think of the right words to phrase this, where someone is in their menstruation cycle, but also like with training and with practice, people can change the way that they smell things. And some of the most popular example, or most noticeable examples, I should say, come from culinary backgrounds, with like smelling of wines and of food and things like that. But people can also be professional perfumers, and that is exactly what it sounds like. People that, for a living, smell specific kind of chemicals and rate them in terms of their hedonic value and how good they are, how bad they are. I'm reminded of wine, for example, that like, this is, I don't know, I'm trying to think about how much into the weeds I want to get on this, because I assume that probably people that are watching this, you've probably tasted wine before, right? And a lot of people, including myself, can't detect that much difference between different kinds of wines. That like, you give me a wine, I can tell you if I like it or not. I can tell you if it's white or red. I can tell you, that's pretty much it, But then you hear about people who are sommeliers, and they've dedicated their life to like wine tasting and to being really, really good at distinguishing wines, grading them, ranking them, telling you all about them. And if you go to a fancy restaurant, you see this stuff, and it always feels like, this guy comes up and he tells you like, oh, this has an oaky bouquet, and it's, it will energize your palate because you can really taste the tannins in this, and it comes from a mahogany barrel or whatever. And I'm always like, yeah, whatever, fine. Do you like it or not? And it feels a little bit like it's bullshit, right? It feels like these people are just kind of like rich snobs just making stuff up. But it is true that people who are wine experts get really, really good at this stuff. And I won't say, that wine experts are better or worse at giving a recommendation for a good wine, because there's been interesting studies that show that, generally speaking, most people prefer kind of the cheaper wines. Your barefoot wines. The wine you can get for $13 or whatever is probably going to be the wine that you like more than one that costs $1,000 per bottle or whatever. But that doesn't mean that there isn't something to their expertise that actually works. That people who are wine experts are actually very accurate about, like, whenever they engage with this wine, they can tell you about the barrel, they can tell you about what kind of mixtures in it, how long it's been prepared, and all this other stuff. It just happens that doesn't always really correlate with what's good and what most people like. But kind of interesting, right? And if you've ever seen anybody who is a sommelier or somebody who is a wine expert, it's interesting they are called wine tasters, because a lot of that experience comes from the smell of it, where they will take a wine glass and they'll kind of, do this little thing right here, and they'll smell it. And that's because wine, the taste is less important than the actual smell in terms of identifying characteristics about it. I was going to say the same thing is true for perfume, but you shouldn't drink perfume. You shouldn't do that. I'm just trying to say that people can get really, really good at identifying and discriminating things just based on scent alone. However, again, this does change over time. By the time we're 85, about half of us are going to be roughly anosmic. And the reason for that could be because of illness. It could also be because by the time we're 85, many of us have fallen down and had injuries where maybe we fractured our cribriform plate and we can't smell, because we have anosmia now. So this changes over time, and you can see here how there is a little bit of a gender divide here again, where we're looking at male versus female, where up here we're looking at higher sensitivities and lower sensitivities, and that women generally have the edge on these over our life cycle. how interesting it is that our ability to smell things, you remember I was talking about how hormones play a part in this, that generally as children we're pretty bad at smelling things and kind of discriminating until puberty, and then all of a sudden we get pretty good at it and we stay pretty good at it until we get older, which again, big changes in hormonal cycles as we get older as well. Okay, let's talk a little bit about adaptation. So adaptation in this context, because the word adaptation has kind of changed a little bit depending on the sense, it means something different for touch as it does for vision, and it means something a little bit different still for smell. Basically, like all of the other senses we've talked about, smell in all of our senses are really, really good at detecting changes. And I notice this all the time with smells, right? Because if you walk into somewhere like a bakery, let's say that you walk into your favorite sandwich shop, Subway, right? You go into Subway and it has that unique kind of bread smell. It doesn't smell like any other kind of bread in the world except for the bread of Subway, right? But it has that very specific kind of smell. Now, if you are in Subway, you walk in there, you smell it immediately, and then pretty much by the time you stand in line and you order and you get your food, you almost don't even smell that smell anymore, right? Because it's not a change. It is not a new thing now. It is part of the environment, and your body's kind of adapted to it and been like, okay, yeah, this isn't anything new. And there's some really interesting reasons for why this might be. Part of it is just through the normal kind of adaptation that our cells go through, where we experience a specific kind of stimulus enough that eventually that sensory system will stop registering it as a new stimulus. And so we don't recognize it as a new stimulus. It doesn't even show up on our radar. The same way that if you hear a low tone, like a radiator in your room, or an air conditioner or whatever, you kind of tune it out over time because it's not new. Same thing for smell. But we can also have receptor adaptation here. And receptor adaptation is essentially like if you are in the subway and you're smelling all this stuff, that those receptors, remember the shape pattern receptors that, take certain kinds of chemicals and will bind onto them, that basically doesn't work because all of those cells are occupied. They all are now, they all have these odorants that are clogging up the receptors, and so we don't detect anything new because all the receptors that are responsible for perceiving bread, or detecting bread, I should say, they're all occupied. And so I'm not getting anything new there. It could also, and this also explains why we have cross-adaptation. So let's say that you go from Subway, where you were just smelling a lot of bread, and then you walk over to Jimmy John's. Do we have Jimmy? I don't know if we have Jimmy John's up here, right, in New England? And if not, then we could say Jersey Mike's. You walk in there, and because that bread is made with many of the same ingredients in terms of yeast, flour, etc., it's not going to register as a new stimulus because a lot of those same receptors are and have been occupied. They have been activated. That's part of the reason why we have this cross-adaptation, where if you are baking brownies in your apartment and then you go next door to a place that I already used chocolate as an example, so I don't want to use chocolate. What's something else that's kind of like brownies? You go to a cookie store, let's say, I don't know. You go to a cake shop. It may not hit you the same way that it would if you just had been on a 30-minute run, or you've been at the gym all day, then you go into this cake store. You're going to smell all this very sweet, frosty type of smells that you're not going to if you already adapted to something very similar, because again, those cells, those receptors have been used. They may still be occupied even with some of those other very similar kind of chemical structures. But we can also have long-term effects of this. And long-term effects of this is called cognitive habituation. And my favorite example of this is anytime you go on vacation, whenever you come back in town and you walk into your house, you immediately smell your house in a way that you don't whenever you get off work, right? So normally, whenever you're coming home from work, you just walk into your apartment, it smells like it doesn't smell like anything. It smells neutral. But if you've been on vacation for 5 days and you come in, now you're like, oh man, it smells like a litter box in here. Or, oh man, it smells like bacon grease in my house. You start getting this new appreciation for what your house normally smells like, and there's a good reason for that. It's because normally we habituate to it with long-term exposure. And there are 3 different factors that can affect this. One is these olfactory receptors that we talked about, that if they get really efficient, then, they adapt very easily, and because of that, that those cells are not detecting a change as easily as they would have because they go through it so much. So that makes sense. This is very similar kind of what we talked about with regular adaptation in the previous slide. The other 2 are a little bit different though. The second reason is because whenever we are detecting these smells, they're getting stuck in our mucous membrane. There's a chance that we can metabolize those smells. That they work their way into our bloodstream. And this can cause us to habituate and to adapt to certain kinds of smells that we are always around. And this would be more of the reason why, like if you work at Subway and you've been working there for a month, when you go into Subway, it no longer really smells like Subway anymore. It just smells like a regular place, much like your house does, because a lot of those signatures are now absorbed in your bloodstream, and so they're part of you now, which is really scary to think about. That you got Subway bread molecules floating around in your blood. It's not that way. It's not as drastic as it sounds. And then there's also cognitive and emotional factors, which could be more behavioral in nature and less biological. There's a really fascinating finding, which is that we can't adapt to smells while we are asleep. There's a very good reason for this, by the way. But I invite you to think about, for me, this makes me think about home. It makes me think about my mom and dad, where I will be asleep on the couch, I will wake up, and my mama has made something awesome that I love. Let's say she made some shrimp and grits. Ooh, wow. I'm so excited for shrimp and grits. I brought that up specifically because I had shrimp earlier today, and I was telling my wife, she's not from the South like I am, and I was like, oh, we got some shrimp. We can make shrimp and grits and have some bacon and some cheese. And she was like, I don't know, I think I'm good. No thanks. But to me, that's a smell that I love. And so if I'm taking a nap on my couch back at home, and my mom's been cooking that while I've been napping for, let's say, an hour, whenever I wake up, I'm like, oh, whoa, what is that smell? It smells great. I can't wait. I'm so, my mouth is salivating now. My brother and my dad, who have been awake this entire time, they don't even smell the shrimp and grits anymore. They've already habituated to it. They've already adapted to shrimp and grits. So even though technically I've been breathing in all those molecules this entire time, it just hasn't adapted for me. So some of these things don't play the same whenever we are asleep. Why is that? Well, it has something to do with our thalamus. So let's talk about our thalamus for a second. Let's talk about how we learn whenever we are smelling things. One of the things that I hear a lot is that smell has the closest link to memory than any other sense. And I feel like every single crime show will have this be an episode. It'll come up in Law and Order SVU, it'll come up on NCIS, it'll come up on CSI, it'll come up on whatever show you like. Maybe not Mindhunter, but it comes up on every show seemingly where somebody is like, oh, and it comes up on House, it comes up on Grey's Anatomy, where they're like, oh, did you know that smell has the strongest link to memory? And that's really, really tricky to kind of assess, because it really begs the question, well, what do we mean by best? Do we mean the most vivid? Do we mean the most long-lasting? Do we mean the most discernible? Do we mean the most discriminable? These are all slightly different questions. Does it mean that's easier to recall or easier to recognize? Because those are also different questions. And so I'm kind of skeptical of this, where I don't want to say that smell memory is better than other kinds of memory. However, there are some key differences that smell memory is unique. I'm not going to say it's the best, but it is unique. And the ways that it's unique stand out in some really interesting ways. So some of it is because odor accesses that emotionality. Remember that when we talk about the olfactory cortex, we talk about the amygdala, right? And the amygdala is the seat of emotion, right? And so that's part of the reason why those memories tied into smell feel so strong. That whenever you smell, let's say that you had shrimp and grits when you were 13, and you got sick afterwards and you barfed all over the place and it made you really, really ill. And so now, whenever you smell shrimp and grits, it makes you sick to your stomach to even to smell it, right? You probably had something like that happen before. We have this taste aversion or the smell aversion. And so we know that like we can have a really, really strong emotional reaction to smell. But does that make it the best sense for memory? I don't think that it does, just because our memories about it are emotional, right? This actually turns out to lead us to one of the problems with smell and memory, which is that we are more likely to make false claims about smells than we are for other types of senses. And the reason for that is because we smell something, we have a really strong emotional reaction, therefore it must be true, right? And this, you see this in a lot of other areas of psychology as well, where people, if they are really emotional about something, they're very convinced about it, and it's because it's very vivid. And people associate accuracy with vividness. They don't always go together. A lot of times they do, but they don't always. And I feel like smell memory is one of those cases where sometimes we might smell things and be very convinced of it because it's very vivid to us and it's very emotional to us, but it's actually not necessarily something that is a better memory. But it may be a longer lasting memory, which only makes this more complicated, right? Because maybe you can have a memory that is long lasting but is not very accurate. And if that's the case, then it's not really the best memory. You see what I mean? Like, it gets really tricky when we start really trying to say, well, what do we mean by the best kind of memory? All right. Part of the reason for this is partly because, and I don't actually mention it here now that I think about it. I should put this in writing because this is really important. That when we talk about, actually, I'm gonna go back a lot for a second here, because I want to show you something when we're talking about the brain and the roadmap to the brain. Okay, so this is where we were, right? So we're going straight from the olfactory sensory neurons to the olfactory bulb, and then straight to the olfactory cortex. We skipped an area. What area did we skip? Every single other major sense goes through this one structure, but not smell. Little structure over here that we call the thalamus. If you remember the thalamus, that's our sensory gateway, right? That sensory gateway mediates vision, it mediates hearing, it mediates taste, it mediates touch, mediates everything. Does not mediate smell. It goes straight to that perceptual area. This is part of the reason why whenever we are sleeping and your thalamus is busy with other things, it's not letting in any of these smells, which is also part of the reason why whenever people are around smoke while they're sleeping, it's not a guarantee they're going to wake up, even if there's a lot of smoke, because the thalamus is doing a really good job of making sure other senses don't make their way, to other areas of the brain. And it kind of picks and chooses what does get there. smell's not even in this category, right? So it's not one of the ones that's being directed all over the place like these other senses. So it's different. And for that reason, people speculate that this is also why memories for smells are exceptionally vivid, is because smell doesn't go through the thalamus like these other senses do. And because it doesn't, that makes it more vivid. That makes it more emotional. That makes it feel more accurate because it's not being kind of regulated by that sensory gateway of the thalamus. So they can be more vivid, they can be more intense, they can be more emotional, but that doesn't necessarily mean that we're more accurate. Other things to mention here, aromatherapy. This comes up a lot. You know, is aromatherapy legit? It seems like there are some things that can influence your body from the way that we smell things. But a lot of time, and I'm talking like, if you know, like scent diffuser or what are they called? Oil diffusers, where they used to be all the hype, like, I don't know, 10 years ago. I feel like they're not as big as they used to be. But these diffusers that would, like, you put in some lavender, you put in some eucalyptus, you put in some citrus or whatever, and it's supposed to change your mood based on that. There's not a lot of great evidence for that to be working, but what does seem to work is that some of these odors we associate with being relaxing, or with being clean, or with being that we associate with being happy, and for that reason, those emotional cues do make us feel a little bit better. So it's not necessarily that that citrus actually makes us feel better or more clear. It's just that we associate citrus with clean, because a lot of cleaning products are made with citrus. And so for that reason, because the entorhinal cortex is such a boss at pairing, associations, that our body is relaxed because we've made these associations. And that's kind of how aromatherapy works. And so there are some very legit kind of effects that we get from smelling things, and that lowering our blood pressure or helping us sleep. So hey, if it works for you, go for it. That's awesome. I'm happy for you. But be a little bit careful with the claims that you're making here, because you know how I am. I'm a little bit of a buzzkill, and I always got to be like, sorry, but that's not actual science, like I was when we're talking about like binaural vibrations that you see on YouTube. All right, let's talk about the final stretch here. Let's talk about pheromones. This is going to lead into our reading, our final research quiz. Our final research quiz is very interested in hormones and whether or not we can detect aggression with hormones. So some of this is going to play a factor into this. Now, a pheromone, if you've never heard that word before, it's used a lot for animal research and to talk about how animals communicate with one another. That a lot of animals communicate not with sound, not with sight, but with scent, right? And you know this because your dogs like to piss on everything, right? And it's not because your dog, is a jerk that he pisses or she pisses on everything. It's because that's kind of how they communicate. That it's a way for your dog to be like, hey, I'm going to let whoever was here know that I was here too. That's all they're saying. I feel like there was, for some reason, we're taught, or at least I was taught, that when a dog pees on something, it means that he's marking it as their territory. I don't think that there's a lot of science that shows that that's what they're doing. But it is a method of communication. It is a way for them to be like, hey, I was here too. Or, hey, I'm ready to reproduce. Because when we talk about animals that are in heat, this also plays a factor of pheromones. So we're talking about a certain kind of chemical scent that we are, that they are detecting and then behaviorally responding to in some way. For a dog that smells another dog in heat, that is going to activate that dog's libido. They're going to get ready to mate. They're going to get ready to have sex. They're going to get in the mood for that type of thing. So it changes their behavior just by smelling something. And there's been a lot of debate about this for humans. Well, does this mean that humans have this? Because it seems like so many other mammals have pheromones and they can communicate with scent. Well, what about humans? Well, we know that pheromones exist in other animals, but the extent that it does for humans is much more nebulous and much less exact. And part of that is because, and I'm spoiling something that's going to come up in a future slide here, that a lot of these animals have a specific kind of organ located in their, what is it called? I'm blanking on the name. It's starts with a V. Sorry, I'm just going to pull this up real quick because I should have put it out on the slide. It is the vomeronasal organ. I was going to call it like the vasal something, so I'm glad I looked it up. The vomeronasal organ. Humans do not have this. At least our understanding is that humans do not have a vomeronasal organ. Other mammals do, other beasts do, but we don't, which means that we don't have pheromones. Or does it? How weird would it be if all these other species do have the ability to communicate with smell, but that we don't? So let's take a quick look at some of the research out here. We know that, for example, monkeys can smell whenever another female monkey is at a specific point in their in their reproductive cycle where they are ready to mate, or will have could have eggs fertilized at that point in their cycles. But if you take a male ape and you block his nose, you put a pin over his nose, he'll ignore that female in heat if that happens, which tells us that it's not anything visual that's going on with this, reproductive pattern. There's nothing hearing, that it has to do with the smell there. Same thing basically for female pigs as well. But what about for people? This is tough because, like a lot of the research, this semester, when we talk about humans, we got to be careful because there are ethical standards where, like, I can't just poke around in someone's brain and see what it does, right? So we have to rely on behavioral evidence for that. And sometimes we can rely on neuroimaging evidence for this too. But it gets tough because we're looking at subjective things now. And so one of the most heavily debated, I would say, out of all of these is the McClintock effect. The McClintock effect, if you're not familiar with this effect, I guarantee you are. You probably just refer to it as syncing. I've heard people refer to it as that their menstrual cycles are in sync. And that's what the McClintock effect is that if you have women that are living together, that generally speaking, over time, their menstrual cycles seem to sync up in ways that do not look like coincidence. It does not appear to be random that this is happening. So, why though? What is that mechanism? It doesn't seem like it would have to do with sight or sound, right? And it probably doesn't have anything to do with touch. So what is it, right? Is it the vibes? Because you're going to need to give me an operational definition for what a vibe is, right? Like, well, what is a vibe? Is it hearing based? Is it, I don't know. It's got to be something, right? And so people have speculated that it might be pheromones. But there's a lot of very mixed evidence on this because Yang and Shank basically tried this where they took McClintock, they took a whole dorm, a whole, what is it called? Not a suite, a whole dormitory of female Chinese students, blocked their noses, didn't seem to stop them from sinking, which was like, okay, well then it must not be smell then, right? If that's true. But again, this is something that like people have replicated this, people have not replicated this. There's a lot of debate about this right now. And so for this very reason, people just call it chemosignals. They're like, okay, well we can't say it's pheromones, we just got to say it's chemosignals. We're not going to use the word pheromones because that must mean that we have a vomeronasal organ. And we don't have a vomeronasal organ because we've seen dead bodies, we've seen brains, they don't have a vomeronasal organ in it, so it must be something else. So a chemosignal is just a very, very broad definition of any kind of signal that we detect with our olfactory system that seems to have an effect on our behavior. That effect may be mood-based, it may be hormonal-based, maybe sex-based, but it's having some kind of effect on us. I'm going to give you two bizarre studies that are not part of our research quiz that kind of look at this. The first is about pregnant women and their ability to detect possibly attractive men. I know this is really, really weird. Let me check how we're doing on time. Okay, I want to get you out of here at the 1 hour 30 minute mark, because I don't like to record longer than that, because that's way too long. So for this study, what they did is they basically brought in a bunch of groups of women, some who were pregnant, some who were not pregnant, and by the way, these are all straight women, cisgender women, and that's important, right? Because we talked about how there are sex differences based on this. And so these researchers used androstenedione, and this is a hormone that is derived from testosterone, and so it naturally occurs in cis male bodies more than females, and you can collect it with sweat. And so the way they did this study, this is wild, y'all. They basically had women look at an array of faces and rate those faces on how sexually attracted they were to those faces. So like attractive face, not attractive face, attractive face, not attractive face, attractive face, not attractive face. And then they gave them a bunch of cotton swabs that had scents of those men, and had them rate, well, how attractive do you think this person is? You know, and they're rating them based on the smell. And what they found is basically nothing. Is that there wasn't really an effect. That, you know, it would be really cool though, right? Like if female experimenters were able to judge, oh, that guy's a hunk. I smell him and he smells great, so he must be really attractive. It turns out there wasn't really a correlation for that, except for pregnant versus non-pregnant women. That the pregnant women were able, better than average, better than chance, at judging how attractive that man was, just based on the scent they got off of that cotton sample, which was part of a sweaty shirt that person had to wear. However, non-pregnant women did not, which again shows the impact that hormonal cycles can play on this. But again, how exactly this works, we still don't really know. And this study was done like almost 20 years ago now. The final one that I want to talk about, and this one is probably not politically correct, so I apologize. But it's science, and this happened. Somebody did this, where these researchers went to these quote-unquote gentlemen clubs, these strip clubs, where basically they would look at how much were these exotic dancers making each night. And they recorded all this over a period of several months. And while they were doing this, they were also looking at whether or not those dancers were menstruating, where they were at on their menstruation cycles, and also whether or not they were on birth control or not. And what they found is that for women that exotic dancers that were in the ovulatory phase of their cycle, so where they stood the greatest probability of being pregnant, huge difference in how much money they made from tips those nights, which is baffling, right? Like, why would that make a difference? It doesn't seem like it should make a difference here, but it's almost twice as much, right? Like, it is a very noticeable difference here. And what they found is that dancers who were on birth control, specifically kinds that were, and I should have done a little bit more research here to find out, because there's so many different kinds of birth control, right? The specific kind of hormone they were kind of controlling for here, but that dancers that were on specific kind of birth controls did not show any kind of fluctuations in their tips. So basically, they maintain the same kind of rate no matter what time of month it was. No matter when it was, they were still making the same amount. Those that were not on birth control overall made more than those that did not, though. And again, you got to wonder, well, why is that? How could these patrons of these clubs that are giving out tips, how do they know? How are they able to perceive this? I don't think you could go to any of these people who are customers and ask them, well, why are you giving more tips to this individual and not this? I don't think they could say, well, this person is ovulating and this one isn't, right? No one would say that. So what is it that they are responding to? Is it something about a visual cue? If so, that seems really hard to imagine, right? Like, how would any of this stuff up here with menstruation cycles have anything to do with visual details, right? Probably doesn't have anything to do with auditory, hearing based. I don't know. People who are skeptics of it, because again, this is all very controversial research and people debate about this stuff all the time. Some people argue that it could be the case that dancers who are ovulating are behaving slightly differently, and those slight differences in behavior are earning more tips. Maybe so. But it could also be something about chemosignals. It could be something about these signatures that we are detecting and unaware of because they're happening at a very low level, but they are subtly changing our behavior to act one way or the other. I don't know. Do you know? If you do, you might make a lot of money and make a lot of waves in the scientific literature by publishing about it, because people are still kind of scratching their heads over this. How is it that we are able to be influenced by these kind of hormonal differences when we don't have the ability to detect pheromones the way that other species do? If you're curious about this, I would invite you to check out at the very, very beginning of this these links about the pheromone parties, because essentially what they do is it's like a blind date, essentially, where instead of being matched up with people at random, you smell a bunch of sweaty t-shirts and then you say, oh, I like this smell the most, and then you're matched up with that person whose smell it was. And I know that sounds kind of weird, but have you ever dated somebody and been in a long-term relationship with somebody you thought smelled bad? Probably not, right? That we seem to be attracted to people that have a specific kind of smell that we like. I don't know. Why do we use perfumes, right? I don't know. It's just interesting, I guess. And so the research article that we have for this chapter or for this unit is about whether or not we can smell if somebody is pissed off, if somebody is aggressive, and if they are mad. Can we detect that based just on sweat? I don't know. You got to find out. I don't want to spoil it for you, but the answer is not as clear-cut as you might think. Thanks for tuning in. I'll see you later. Bye.